you should have Googled that in gigahertz, KBT at room temperature is about, I think, 6,000 gigahertz or something. And one question that I don't usually get for NV centers, but that I usually get from what I'm going to talk about now is what happens now. So your gigahertz, uh, your qubit frequency is much smaller than KBT. So how can you use it as a quantum sensor at all, right? So the answer to this, and it's important that I mention this now, is the fact that yes, KBT is much larger than the qubit difference in energy, but all the quantum information that we're able to obtain with the sensor is only coming from times before the qubit comes in equilibrium, thermal equilibrium with the environment. The time that it takes for the qubit to come to equilibrium with thermal equilibrium to the environment is about, in this case, more or less without any control pulse, about 2 microseconds. Okay, so it is possible to obtain quantum information from sensors that have a transition frequency much smaller than KBT if the information is gotten while the thing is in a proper quantum state, is well described by uh, the laws of quantum mechanics. Okay, and you're going to see why I wanted to mention this important thing right now. So now I'm going to the second part of my talk to talk about what I really want to talk about. So before in my life, those were the types of sensors that I was used to working with. But at some point, I realized that, well, nature is a very good engineer, right? It has had millions of years to perfect their sensors, and in many cases, natural sensors overperform humankind made sensors in crazy ways. In my past, I've worked with uh, two types of natural sensors, a light sensor that is not quantum at all, and a magnetic field sensor that I'm going to argue uh, seems to be quantum. Okay, so let me talk to you about this natural quantum magnetic field sensor. But in order to do so, I'll have to start with biology at the nanoscale, namely the chemistry. So it's been known in basic chemistry that for decades now, that magnetic fields can alter the final products of a class of chemical reactions that involves spin correlated electrons. And yes, by spin correlated electrons, I mean that they are entangled. Okay, so let me tell you how this works uh, in chemical reactions. Again, no no doubt that this is what happens in the lab when chemists are playing with compounds. So imagine that. So and now you should be looking at me. I have my props. I carry my spins around. So imagine that you have two spins at the outer shell of a, say, fluorescence protein, or like a chromophore, something that can be excited, and as it excites, it emits light, and this light is known as fluorescence. So those two spins, they are in a singlet state, one pointing up and one pointing down, just because either of the Pauli exclusion principle, or you can think about it in this because that's their most energetically favorable state. So if a photon comes in, it gives enough energy of appropriate frequency, it gives enough energy for one electron to be promoted, and most times this electron will come back down, emitting fluorescence. Sometimes though, upon absorption of a photon, this guy here will be excited, and before it excites back, sometimes it meets with another electron spin in solution, a free radical. Free radicals are very common to find in chemical reactions. Okay? If those two spins meet, okay, the first point that I want to make is that because photon absorption processes are spin conserving, whenever this guy meets with this guy, they need to start in a singlet state because those, in the absence of any other laws, because those two spins were in a singlet state. So I hope you agree with me that in the absence of laws, those two spins need to start in a singlet state. Well, singlet states, people sometimes we forget that singlet states are spin correlated, are entangled. So it's in this sense that this mechanism depends on entangled electrons. The second thing that you need to know is that there are chemical reactions that are spin dependent. This means that the chemical reactions, they continue through one branch if the spins are, and now it's jargon, measured by the environment to be in a singlet state, and the chemical reaction continues through another branch if the electron spins are measured by the environment statistically to be in a triplet state. Importantly, the final products of those two branches are macroscopically different. Okay, so a putative, fast quantum mechanical phenomenon that happens at the top of a chemical reaction where electrons can be found either like this, or they can alternate. A fast process occurring at the top of a chemical reaction that changes spins can have macroscopic signatures downstream at much longer timescales. Okay? Third thing that you need to know is that in this case, those two electron spins are actually have different local magnetic field environments. So this electron here, it's usually coupled to nuclear spins with the molecule where it came from, whereas this electron spin here, it's free. So in the presence of an external magnetic field of a strength that is small compared to the hyperfine interactions, this guy will Larmor precess around this Larmor frequency of the external magnetic field, whereas this guy will Larmor precess around the frequency of this external magnetic field plus the Larmor frequency like of this hyperfine interaction, which sort of dominates. Okay, so in the presence of a weak magnetic field, this guy is doing its thing. And if this external magnetic field shifts a little bit, this guy doesn't care, but the Larmor frequency of this guy is strongly altered. This means that actually, upon tiny changes of a weak external magnetic field, those two spins are going to pick different phases with respect to each other, all because the Larmor frequency of this guy uh, changes well, changes by a lot. So the guy who's doing all the job in picking up, so that those two spins, as they Larmor process, pick up different phases, this guy is the one doing all the job. This difference in phase actually translates into a difference in probability that the spins are measured by the environment to be in a singlet or in a triplet state. All I'm saying is that an external weak magnetic field or changes in it is going to alter the probability that the spins at the top of a chemical reaction are found in a triplet or singlet state. So effectively, an external weak magnetic field is going to alter chemical pathways in chemical reactions, can alter in specific molecules. Okay, so this is known in chemistry, and all the uh, ways to explain this pass through a dependence of this process on the spins of involved. Okay, there is no alternative phenomena. There's no alternative explanation to what happens in chemical process. Okay? So, and now, even though this is not a bird talk, I need to start talking about birds because in the 70s, many years ago, biophysicists, theoretical biophysicists, they wanted to understand how birds actually are using the magnetic field of the Earth to migrate. There's no doubt that birds, when they go north to south, they use the magnetic field of the Earth at least as a partial cue. And again, the magnetic field of the Earth is about 50 micron Tesla, which is weaker than the magnetic field that you feel when you put your cell phone close to your head. So how might they be doing this? So the theoretical biophysicists, they propose that were the same type of spin dependent chemical reaction at work physiologically inside the bird somehow, right? Birds could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from this sort of spin dependent chemical reactions. And if that were the case, this would mean that there is a transient, coherent uh, process that is spin process that would be happening inside the bird. And at that point in time, the only and because this process that I just described usually starts with the absorption of a photon, right, that excites this guy. Scientists, biophysicists started looking for molecules, biological molecules that absorb light. So the first thing that they looked at were photoreceptors, and at that time, the only known animal photoreceptors uh, known to sustain, to be able to sustain such a spin-dependent chemical reaction was a photoreceptor that is present in our eyes, in the eyes of migrating birds, in the antennas of migrating butterflies. It's a protein, a photoreceptor called a cryptochrome. Cryptochrome is a protein that has a flavin. The flavin is the thing that makes it fluoresce. Uh, Again, it's a photoreceptor, but interestingly, it's present, and I think this statement is accurate. It's present in all our cells because it also has circadian rhythm regulation functions. And actually, and this is not weird science. The reason why when you're jet lagged, people say, "Oh, stare at blue light," is to sort of try to change a little bit the workings of your cryptochromes that absorb in the blue uh, spectrum. Okay, and it turns out that there beyond birds there are many uh, organisms that seem to be magnetosensing via ways that seem to depend on cryptochrome. First of all, cryptochrome is absolutely universal across the tree of life, from microbes to like humans, right? To plants, to reptiles, to birds. Um, and so in this slide there, those are some organisms that express cryptochromes, and boxed in red are organisms uh, that studies, there have been magnetosensing studies that have shown some, sometimes weak, but correlation of magnetosensing with cryptochrome. And yes, humans are right there. And actually, evidence for cryptochrome-based magnetosensing is really widespread, but unfortunately at disconnected length scales. Okay? In the very tiny length scales, and it's in the left part of my slides, there are chemical studies on cryptochrome in solution that sort of show that cryptochrome does work as a quantum sensor of magnetic fields at room temperature. Let me go through some of the evidence that you see there to your left. Okay, so in one study, researchers captured the fluorescence emitted by cryptochrome as a function of the time during which you excited cryptochrome with an appropriate frequency that could make spin transitions. Okay? The first thing that you see, this is for cryptochrome solution. The first thing that you see is that this cryptochrome is emitting fluorescence, but this fluorescence is going down. This means that you're killing sort of your sample by applying very strong laser fields onto that. But as the researchers pulsed a tiny magnetic field on and off as they were capturing this fluorescence, and this is not an artifact. They have tried different magnetic field on and off frequencies, intensities. What they see is a tiny fluorescence modulation as the fluorescence is decaying, right? The fluorescence comes up and down. I'm going to argue that this is an analogy with the single spin in diamond. For the single